So this is an overview of your readings for Chapter 21. This is the evolution of populations. And it's going to cover genetic drift, natural selection, Hardy-Weinberg, and genetic flow. So because there's a lot of words and diagrams, I'm going to get rid of the video feed for this episode just to have a better view for you guys. Alright, so we're going to start with microevolution. Microevolution is only talking about the change in allele frequencies of a population over several generations, and it's the driving factor for macroevolution, which is the larger scale what we eventually would see. Microevolution is only dealing with the alleles. If you can remember, the alleles are 1/2 of the genotype. So if I have big A, little a, those are the alleles. Okay, that's what you inherit from your parents. Now, three things can cause your genetic change. One, natural selection, genetic drift, genetic flow. Okay, but only natural selection caused the adaptive evolution. These other twos will either get rid of an allele or introduce an allele. So we're going to start with the Hardy-Weinberg equation just because it's a little bit easier to see the mathematics behind it before actually going into the whole thing. So a population is a localized group of individuals capable of interbreeding, there's the key, and producing fertile offspring. They have to be able to produce offspring that are able to produce offspring. Otherwise, the population is just going to die. So, for example, mules. Mules are the result of a donkey and a horse, but they can't produce fertile offspring. In other words, you can't mate two mules. They just don't have the equipment for it. A gene pool is all the alleles for the population. Okay, so what it means by this for all loci in a population is that the population has on their chromosomes different places where the alleles are. And that's just talking about that. So gene pool is all the alleles for that specific gene in a population. Okay. If we look at this picture, this is horses of several different colors. We have what we call a sorrel, a bay, and a brown. They've got different spots, different markings, stockings. They all are the same species, meaning they can all interbreed, and they all have the same gene for these colorings, just different alleles, giving them different colors. Okay, so these are what we call phenotypic variations. They look different. Phenotype is what we see, it's their physical traits. Well, the physical traits, different colors. For our Hardy-Weinberg equation, Hardy-Weinberg, they were two different scientists that came up with the same mathematical equation for determining if a population is evolving, better yet, is not evolving. Because if the Hardy-Weinberg equation is met, then the population is said not to be evolving. And that's what this says. Hardy-Weinberg principle states that frequencies of alleles and genotypes in a population remain constant from generation to generation. In other words, non-evolving. To be non-evolving, their populations have to be no mutations, no random, or they have to have random mating. You cannot selectively mate two pairs. It has to be random. No natural selection, large population size. And by large population size, we're talking in the upper thousands, okay? It can't just be 40, it can't just be 100, it's got to be practically above 1,000. And then no gene flow. This means no new genes coming in or no new genes going, or old genes going out. For example, migration or extinction. The Hardy-Weinberg principle describes that population that's not evolving. Okay, if it does not meet that criteria, then it's concluded that the population is evolving. Okay, so this is how scientists use a determining if a population is evolving or not. Here's the equations, okay? You're going to have to memorize these two. And very, very easy equations, and they're applied in a similar way. So, if P and Q represent the relative frequencies of only two possible alleles in a population at a particular locus, in other words, for one gene, then these two things are going to they're going to work, okay? So if we have half of the gene pool is big A and half of the gene pool is little a, that'll be 0.5 plus 0.5 equals 1. Okay? Then we can use this side to determine what the genotypes are. If you remember, there's homologous dominant, homologous recessive, and heterozygous. Okay? So, if we look at a population and we see that there are 25% of the population is homozygous recessive. That's the recessive gene. You have to have little a, little a. We're going to assign that 25% or 0.25 to the q squared. Okay. Then we solve for p. Well, we know that this is 25% or 0.25. We can plug that q squared, figure out what q is from that. So the square root of 0.25 is 0.5. Go back to this equation and say 0.5 minus, or 1 minus 0.5 is 0.5. Now we have our p, and we can figure out what the other two possible genotypes are. Okay? Using that equation, we can determine if a population is staying constant and not evolving, or more likely it is evolving. So three things can alter these this Hardy-Weinberg, okay, and it's natural selection, genetic drift, and gene flow. So let's start with natural selection, the one you're more commonly familiar with. It's a change of genes from generation to generation. For example, if I spray plants with DDT to get rid of some pests in there, the next generation, well, there's going to be some more pests come back because they've become resistant to it. And that resistance will increase in frequency over time because those insects are passing along their genes. Okay, so it's success in reproduction results in certain alleles being passed to the next generation. Okay, and it keeps increasing over time. Genetic drift is a little bit different in that genetic drift is you have to have a small sample if there is a small population. Randomly, the genes will increase or decrease over time. And this is completely random, it's unpredictable, and that frequency fluctuation from from generation to generation will change what the population looks like. Okay, so it'll evolve to either have more of something or have less of something. I'll show you a couple examples of these, but we're going to talk about two ways that this happens in nature. The founder's effect, which is when a few individuals become isolated from a larger population. Let's imagine on the Galapagos Islands, a few finches flew from one island to the next. They became founders or pioneers. They passed on only the traits that they had. They did not carry any of the other traits, so they weren't able to pass on any of those other traits. The population on that new island grew, but it only had the alleles that those founders had. It cannot just randomly develop something else, unless there is a random mutation, which is very rare. You could also have a bottleneck effect. This is what happens. It happened in cheetahs and it happened in Florida panthers. Usually there is some sort of man-made or disaster that reduces the population very, very low. This population reduces the gene pool, and only the alleles left are passed on. Very similar to the founder's effect. Okay, so here's a diagram showing that. We have the original population. There's several different alleles, a few random ones in there, and it gets bottlenecked, or the population is reduced. The only population surviving does not have these yellow beads in it. In other words, that yellow bead is now extinct and won't show up in future populations. There's our Florida panther. Okay. All right, so genetic drift is significant in small populations. If you have a large population, you don't have to worry about genetic drift as much. Genetic drift can also cause allele frequencies to change at random. We don't know, we can't predict which way it's going to go. And it can lead to a loss of genetic variation, as seen by the founder's effect and the bottleneck effect. And genetic drift can cause harmful alleles to become fixed. What I mean by fixed is always present, or the only allele left. Okay? This is very dangerous for certain populations and can lead to extinction. Our last one that we need to talk about is gene flow. Gene flow consists of the movement of alleles among population due to mating. So this can be selective mating. This can be mating chose by the animals themselves or by mankind. Alleles can be transferred through the movement of fertile individuals or gametes, and the gene flow tends to reduce genetic variation among populations over time, as seen by purebred dogs. So man chose which dogs he wanted to interbreed, and over time those genetic variations among those dogs decrease significantly. They all look the same. Okay? Gene flow can decrease the fitness of a population. So like I'm talking about with the dogs, it's not, those purebred dogs tend to have a lot of health issues, and so they suffer from anywhere from hip dysplasia to bad gums and bad teeth and slipped vertebrae. Another example is the bird the great tit, which is Paris major, on the island of Valand. Okay? Immigration of certain birds came from the mainland. They introduced alleles that decreased fitness in island populations. In other words, we introduced certain things that decreased populations on these islands. Natural selection reduced the frequencies of these alleles in the eastern population where immigration from the mainland was low. So it caused a problem from others on the other side of the island. But in the central population, high immigration from the mainland overwhelms the effect of selection, causing the other birds to die off. All right. So evolution by natural selection is involving both chance and sorting. That's where the animals choose. New genetic variations arise by chance, but beneficial alleles are sorted and favored by natural selection. If you're stronger, faster, better, well, your mate is going to look for you. And they're going to choose you over the one that's not so well. Only natural selection consistently results in adaptive evolution, which is an increase of frequency that improve fitness. They help the animal. So that's natural selection. It's still random because those new genetic variations are only by chance, but once they're there, they improve fitness, which causes them to improve the entire population. Okay, there's 3 ways that this can happen. So there's directional selection, which is going to kind of move the entire population one way, making them better overall. There's a disruptive selection, which favors individuals at the extremes of the phenotype. So if you have several different colors of skin, well, disruptive selection favored the really fair people and the really dark people. And therefore, we now have a disruptive selection. Eventually, what that could lead to is a separation of species if there was not interbreeding between them. Stabilizing selection is they're going to favor the people in the middle or the animals in the middle. This is getting rid of those extreme examples from the sides. Here is a example of that. Okay, so if here's our population originally. Okay, we have a light mouse varying all the way to a dark mouse. If we were in a dark environment, what would happen is directional selection where only the dark mice would survive because they could hide easier. Okay, a disruptive selection would be where only the light and the dark survive, not the mid-range. And then the stabilizing selection, more of the population becomes the middle mid-color. Okay. All right, that's all for this readings. Hopefully you were able to get through this. Hopefully this video helps you some. I will be back in on Tuesday to kind of go over what you've missed and start going on for further and further and further.